If we go back to a blank slate, and you start with NAD, we're supposed to get 2 ATP, right? Well, 2 protons are pumped here. 2 protons are pumped here. If these go through, 1 ATP. When these go through, another ATP. Therefore, we get 2 ATP. Okay? So the whole purpose then of this electron transport, the whole purpose of moving those protons into that intermembrane space, we can say, is that it's to maintain a concentration gradient. The whole purpose of collecting the energy in the form of NADH and FADH2 from glycolysis and the matrix reactions is that energy is then used to maintain that concentration gradient. So we can continue to have protons moving through the ATP synthase, generating ATP continually. But here's the problem. What if we don't have oxygen, or the oxygen supplies run low? Remember, oxygen acts as a final electron acceptor. If we don't have oxygen, there is no place for this final cytochrome, A3, to send its electrons. It stays in the reduced form. If the electrons are still attached here, that means that any electrons here stay, and any electrons here stay, which means that additional NADH are unable to unload their electrons, and additional FADH2s are unable to unload their electrons. So you get this backup. This assembly line essentially stops. No more protons are pumped into the intermembrane space. Eventually, what's going to happen to that proton gradient? It's going to decrease. Eventually, as protons keep moving through here, because that's what they want to do, because they're more highly concentrated here than here, as they continue to move through here, yeah, they'll generate ATP for a short time. And I'm talking very, very short from a cellular perspective, very short period of time. But because there's no other protons being pumped up here because everything's backed up, eventually you're going to reach an equilibrium and you're not going to have that gradient anymore and you're not going to be able to produce ATP. Now we're talking hundreds, maybe thousandths of a second. So in our span of time, it's instantaneous. Cellular time, it's fast, but not instantaneous. That's because oxygen is not present to form the water with the protons. That's the only way that those electrons can get to the protons, which are in that reduced form. So without the oxygen, everything stays. If this is full, you can't hand the electrons back off. So everything stops. And eventually that gradient slows down. You reach an equilibrium. You don't get the ATP produced. Now, there are mechanisms in place within the cell that do allow a little bit of ATP to be produced. Not a lot, but a little bit of ATP to be produced to keep the cell going until there's enough oxygen available for this process to continue again. Now, you've heard of cyanide poisoning. The way they portray spies in the movies where they bite a cyanide capsule and they die instantly, that's not real. You can't have a guy in a movie for dramatic effect waiting for 5 minutes for him to die after eating a cyanide capsule. So the dramatic effect in the movie, the creative licenses, they die instantly. No, not really. What cyanide does is it blocks the transfer of the electrons from the cytochrome A3 to oxygen. It prevents that from happening, even when oxygen is present. So if you can't move those electrons from the last cytochrome, that whole transport of electrons down that chain stops. And then eventually that gradient runs out. ATP can't be produced. For a short period of time, the cell can produce small amounts of ATP anaerobically, but that doesn't last forever, and eventually the cell doesn't produce ATP, and the cells die, and when all the cells die, you die. If anybody's looking for 2 more points of extra credit, Maybe do a little more detailed research on cyanide poisoning. Don't email it to me. I will put a link on Blackboard for you to submit it. But that might be something you can do. Looking specifically beyond the fact that cyanide blocks the transfer. How then does that cause death? So if you want to go back and look at totals again, I'll have to reduplicate my chart here. I'm not sure why I don't have it as a table in here anymore. Notice that on the bottom of that table it should say FADH2, not FAHD, FAHD2 or whatever, the transposed. So if we take a look at this, let's go back and review. Glycolysis. How much ATP did we use? 2. How much did we produce total? 4, but the net gain of ATP is 2. How much NADH did we get? 2. FADH2? 0. All right, in the matrix reaction, did we use ATP? No. Did we produce ATP? Per glucose, how much ATP did we produce? 2. Per pyruvate, it's 1 in the citric acid cycle, but we have 2 pyruvic acids for every glucose that have to go through that reaction. So net ATP is 2. NADH, 8. FADH2, 2. Now in the membrane reactions, we don't use ATP, but we do produce a lot. Do we produce any NADH? No. Do we produce any FADH2? No, but they do undergo oxidation. So these 10 here in the membrane reactions will yield approximately how many ATP? 30. And these 2 FADHs here will yield approximately 4 ATP. So when we add up the total ATP from the membrane reactions, it's 34. Our total ATP produced then, 2 plus 2 plus 34, 38 ATP. So when the NADH gives up its electrons, it then becomes oxidized to NAD, right? It also gives up its protons. So there's 2 things that can happen to the protons. They can be pumped up here, or they can be used as the protons that are part of forming water. Doesn't matter which. They just get added to the proton pool in the matrix.